In this video, you could see how to obtain skeletal elements from some marine invertebrates. Objectives of this session are, understanding the importance of assessing biodiversity variability, increasing the ability to observe and to learn from nature, understanding how to properly label samples, learning how to handle samples and lab tools. First examined species is the Gorgonian, Leptogorgis hermatosa. Gorgonians form erect and branched colonies. Living colonies resemble hairy, due to numerous, small individuals called anthocodia or polyps. This anthozoan is an octocoral, due to its eightfold radial symmetry, and because its polyps presents 8 tentacles. These tentacles show numerous, tiny side branches called pinnules. The living tissue of Gorgonians is called synenchyme. The synenchyme can have bright colors due to pigments. Leptogorgis hermatosa generally is pink to orange. Gorgonians are supported by an inner skeleton mainly composed of proteins that make colonies flexible. This limits colony breaking due to strong currents in marine environment. The living tissue is embedded of skeletal elements called sclerites made of calcium carbonate. Sclerites are embedded into mesoglea. They generally measure less than 1 mm, even if they can be longer than 1 mm in some species of Gorgonians. Sclerites may have different shapes and functions in relation to species and to the considered portion of the Gorgonian. For example, sclerites at the base of the polyp are different from those present below tentacles. First function of sclerites is structural. Together with the core axis, sclerites support colonies. They are arranged in layers around the axis. Sclerites can be oriented in different ways to give resistance and flexibility to colonies. It is necessary that colonies were flexible to avoid breakage due to water movement. The arrangement of sclerites embedded into mesoglea can vary depending on the forces acting on the Gorgonian. Sclerites embedded into mesoglea can do slight movements to contrast compression and extension of colonies due to water movement. The mesoglea itself is elastic and acts synergistically with sclerites to define freedom of movement in response to external forces. Sclerites are used as important taxonomic character to distinguish Gorgonian species. However, size of sclerites within a species may change in relation to environmental factors. For example, due to intensity of water movement or depth. Therefore, it would be relevant to analyze sets of sclerites from Gorgonians belonging to the same species but collected in different localities or different bathymetric ranges. In order to obtain and to study a set of sclerites, you could use both dry or wet samples. In these clips, you will see a dry specimens of Leptogorgis hermatosa from shallow waters of the Adriatic Sea. How to obtain sclerites. Method 1. Quick method. First, cut a small fragment from the colony. Take note of which portion you are considering, since sclerites from the basal region could be different respect to those present in the apexes. Introduce the fragment inside a tube containing hydrogen peroxide or sodium hypochlorite. These reagents will digest the living tissues, while the sclerites made of calcium carbonate will precipitate to the bottom of the tube. Digestion time could depend on 1. Concentration of the reagent, 2. Thickness of the After a while, the tissues of the Gorgonian will be digested, and sclerites will sink toward the bottom of the tube. If not much time was passed from the beginning of the reaction, it will be still possible to observe the core axis. Now, it is necessary to rinse the sample, to eliminate the reagent. Throw out the reagent in a jar, being careful not to lose the sclerites. Remember to follow appropriate disposal procedure for any reagent. Rinse the sample by introducing in the tube distilled water. In order to rinse well the sclerites, and avoid the formation of artifacts, shake the tube, or, use a vortex mixer to resuspend the sclerites. When all the sclerites precipitate toward the bottom, and the supernatant is clear, repeat rinsing one more time. After rinsing, left few drops of water inside the tube to resuspend and to collect the sclerites from the bottom of the tube. Collect the sclerites with a pipette. Put the sclerites on a slide. Take a cover glass with forceps, put an extremity of the cover glass on the slide, then, slowly tip it onto the slide to limit the formation of bubbles. How to obtain sclerites. Method 2. Slow method. First, remember to label tubes and slides, especially if you have to manage many samples. When samples are labeled, it would be preferable to use smart codes. Smart codes include unambiguous details about a sample. For example, in the code, Ms. ASEAN November 18, 2015 P001, the first, two-letter code is the Alpha 2 code indicating a country where the sample was collected. The second code is specific for the considered taxon. Then, there is the sampling date. The last code, in this case, PO, refers to a locality. At the end, there is a progressive number to distinguish samples collected at the same place and on the same day. As seen previously in the quick method, cut a portion of Gorgonian and put the fragment inside a tube. Introduce into the tube sodium hypochlorite 5%, filling about half volume of the tube. In order to speed up the digestion of the organic matter, it would be possible to use sodium hypochlorite and heat. In this case, remember to use glass tubes. The procedure to obtain sclerites from morphological studies could be carried out simultaneous to that to obtain the abundance of the sclerites expressed as percentage weight of sclerites on weight of Gorgonian. Determine the weight of a lab tube using an analytic scale. Record all the measurements on a spreadsheet. Determine the gross weight, tube plus a fragment of Gorgonian. The weight of the fragment of Gorgonian, or, net weight, is given by the difference between the gross weight and the tare. After digesting, rinsing and drying the sample, determine the gross weight of the tube containing the sclerites on the bottom. Obtain the tare subtracting the weight of the tube, then, calculate the percentage weight of sclerites on the weight of the Gorgonian fragment. The next clips show the explained passages of weighting and boiling samples. Here, the researcher is using a Bunsen burner to boil a tube containing sodium hypochlorite and a fragment of Gorgonian. When you are boiling a sample, do not keep the tube too much time on the Bunsen, to avoid breakage of the glass. When the sample is boiling, it would be possible to see foam. During this passage, the reagent digests the organic matter, while the inorganic fraction, represented by sclerites, remains on the tube. After about 5 to 10 minutes, the organic matter should be degraded. Before going ahead with rinsing, wait until the tubes are cool. After 2 passages in distilled water, rinse with at least 70% ethanol. After each passage, wait for sclerites sediment toward the bottom. Then, throw away the ethanol, and dry the tubes inside a muffle furnace at about 60 Celsius degree. At this point, it will be possible to weight the completely dried samples. The same sample could be used to obtain slides to be observed by light microscopy. Resuspend the sclerites in a few drops of ethanol, then, collect them by a pipette. Put the sclerites on a slide and wait for the ethanol to evaporate. You could put the slides on a hot plate to speed up the evaporation. At the end, put a mounting medium on the slides, therefore, close the sample with a cover glass. When dry, this will be a permanent slide. Now you can observe your slide and measure sclerites.